Welcome back, kids and coaches, and Dr. Mike here again. And today we are getting into learning outcome 2 out of unit 2 of FW-250. So today what we're going to be doing is learning about more of the microanatomy of our muscle fibers and then talking about the sliding filament theory. So let's go ahead and break this down from a fundamental standpoint. Now, just as kind of a little bit of an overview here, we last spoke about the overall structure and anatomy of the muscle itself. So I want to start with a little bit of a refresher on that. So when we're getting into the microanatomy, we're talking about everything that is a single muscle fiber and smaller. So our muscle fiber is made up of component parts called the myofibril. So this is again the kind of rod-like components that are made up really of proteins. So this is where you're going to find like your actin, myosin, your titin, and this or many of these together form our actual muscle fiber itself. Now, when we look at the myofibril, this gets broken down further into sarcomeres. Sarcomeres are the component parts that make up myofibrils. So many of these sarcomeres together will add up to make one myofibril. Now, we also discussed how the sarcolemma here is really just the cell membrane of our muscle fiber. Inside that is where we find the myofibrils. We'll also find our mitochondria, and then the substance inside there. So all of this in here, this is all your sarcoplasm. Inside the sarcoplasm, you're going to find glycogen. So that's where our actual intramuscular glycogen is housed. Now, when we get here into the anatomy of a myofibril, that's where we get into the place where we start finding out how actual muscle contraction occurs. So all of our contractile elements are made up of really just proteins. So when you break down a myofibril, all you're talking about is what the component proteins are that actually go into making a muscle contraction possible. Now, there's a few main ones that we should remember. The two biggest ones are actin and myosin. These are known as our thin and our thick filaments. These are the two proteins that actually cross over each other to make a muscle contraction occur. And we'll talk about that on the next slide when we get into the actual sliding filament theory. We also have, which is not pictured here, but titin. Titin's a giant protein that we have, and by giant protein meaning that it's about 1 micron or 1 micrometer in diameter. To give you some reference, a regular human hair is about 20 to 200 microns or micrometers in diameter. So still pretty big if you really think about it. Now, titin just connects our Z line to the M line. So what this just does is it binds myosin. So our thick filaments is our myosin here. So you have the Z line here, and titin will bind that myosin to that. So that's all it really does. Myosin, you can also think of as making up the A band, And actin, you can think of, is making up your I band. Now, a couple other things we should be aware of here. We have this thing called the H zone. We have our Z line, which again is going to be about right there. And then we have our M line. Where our Z line is, this is where we have our actin binding to this. So the Z line, and in here it's labeled Z discs, this is where the actin is going to actually hold itself. When you have two Z lines together, so two Z lines back to back, so here's a Z line, here's a Z line, here's a Z line, every two Z lines make up a sarcomere. Now our H zone here, this is just where the A bands are present, so where your myosin is present, but no actin is available. So your H zone is just where the myosin is and the actin isn't. And then your M line, you can think of your M line is colloquially known as the middle line of a sarcomere. So an M line is the middle of the sarcomere. What's happening here is this is where your myosin is going to attach, or where it originates, and then your myosin is going to go off in both directions. Eventually, the myosin is going to attach to your Z line via titin. So your M line is just the middle portion of the sarcomere. This is where your myosin originates. And from here, the myosin reaches out in opposite directions to try to grab onto your Z line. Now I do have here the mention of troponin and tropomyosin. What these are pretty much, I mean, they're more proteins here. Troponin attaches to tropomyosin, and these are going to be really present in between the actin. So tropomyosin is important during the relaxation phase of a muscle contraction. So when your muscle is not contracting, it's just relaxing, the tropomyosin is going to prevent myosin from attaching onto the actin. So this is what actually prevents you from having a muscle contraction. When you do have a muscle contraction, troponin will pull the tropomyosin away to allow that myosin to attach to your actin. So these are actually going to be located, if you can see here, in these little grooves that we see in our actin. So that's where you would find your troponin and tropomyosin. Now, I want to finish off here with our sliding filament theory, because this is what actually tells us how we go about contracting or making a muscle contraction. What this is explaining here is the process on the protein level that occurs when muscle contraction actually happens. In short, when you contract a muscle, all that happens is your actin and your myosin attach. When those two attach, you have a muscle contraction. So right here is your actin, right here is your myosin. When you first start this, so this is breaking it down into a 4-step process. But when you first start the sliding filament theory, you notice that the actin and myosin are not touching, they are apart. So that's perfectly fine. As long as they stay apart, we do not have a muscle contraction. Now, when you do start this process, what will happen here is that we get the head of myosin attaching to the actin. This forms what's known as a cross bridge. So a cross bridge is just where you have an intersection of your actin and myosin. So you notice here, these lines all overlap each other. That is what your cross bridge would look like. After we get the myosin head binding to our actin, we get what's known as the power stroke. And what's happening here is that your myosin head actually is going to rotate towards the center of the sarcomere. So if I clear out all the drawings here, you can see that this head starts up, virtually up and down. Here it's at a bit of an angle, and now it's almost straight across. So you can see how that head placement has changed as we go. When this happens, this is when we have the actual forceful muscle contraction occurring. So as long as the myosin head stays attached to the actin, we get our actual muscle contraction. Now, what happens here is that we get the presence of ATP, or adenosine triphosphate. What's happening here then is that as your myosin heads are binding or attaching here to the actin, we have to get ATP on there. And the ATP is the energy we need to remove the muscle contraction. Once you contract a muscle, you don't need ATP to keep it contracted. What you need is you need to have calcium to keep it contracted. So when these two attach to each other, what's actually happening here is calcium is the one that holds that together. So as long as you have calcium present, you'll keep this cross bridge intact. When ATP comes around, ATP attaches to the myosin head and it breaks up the party. ATP is the jealous girlfriend here because she sees that the actin is hanging out with the myosin and she does not want myosin to be with actin. So when the ATP comes about, it breaks up this party. So ATP is required to make actin and myosin release or detach from each other. This comes with a bit of a caveat though, because you need to get the calcium to release as well. If the calcium won't release, the ATP is not allowed in. If calcium stays bound to the actin-myosin cross bridge, you cannot separate it. Under normal circumstances, under a normal muscle contraction, the calcium naturally goes away from your muscle or away from the cross bridge as you relax the muscle. But for any of you that have ever had a muscle cramp, you know the muscle contracts and it stays contracted even when you don't want it to be contracted. That's because the calcium is not able to release the cross bridge to allow ATP in. Now, we'll get into what's known as your maximum voluntary isometric contraction. This is how much or how much force your muscle can actually provide you. And we'll see that your muscles cannot contract at 100%. Your body regulates that because your muscles are a lot stronger than what your body can handle. So your body has to actually down regulate them, meaning it puts a governor or almost like a restrictor plate on there for all you NASCAR fans. And it makes it so that you can't contract with the full force of your muscle because you would risk hurting yourself. To get even close to 100% MVIC or maximum voluntary isometric contraction, you have to have first a muscle cramp. And anyone that had a muscle cramp, you know that those don't feel good. The only way to get 100% of your MVIC is to have a little something called tetanus. If you have tetanus, colloquially it's known as lockjaw, calcium becomes permanently bound here and you are unable to relax that. So tetanus is caused by a pathogen. It's the tetani bacteria. And when you get this, when you're infected with this, it's typically because you've gotten, like I say, a cut or some sort of puncture wound from a metal object, because that's where it likes to live. You usually go and get a tetanus shot. If you didn't have immunity to this and you got tetanus, you would get 100%, eventually you'd get 100% of your maximum voluntary isometric contraction, and your muscles would be permanently contracted in full force. Not only would you not be able to move at all, but it would be extremely painful. So one of the worst ways to die, I would think, would be via tetanus because of how just agonizing that pain would be going on there. So just to recap, we start out here with actin and myosin being close to each other, relatively speaking, but they don't touch each other. You get calcium to bind to the actin and myosin, causing a cross bridge to form. So the actin head bonds to the myosin head bonds to the actin filament to cause a cross bridge. After that, the myosin head rotates in what's known as the power stroke, which causes the actual muscle contraction that we see. To then relax, after we do our muscle contraction, ATP enters and it breaks up the cross bridge. And then our system keeps recycling here. At this point, our calcium also leaves. So that there should give you a better understanding of the microanatomy of our muscle fibers, as well as the sliding filament theory, or how we actually make muscle contractions. So until next time, I hope you have all learned something, and I will catch you guys all later.